Another organic synthesis strategy is to make a carbon-carbon bond to make a bigger molecule from a smaller molecule. To make a carbon-carbon bond, we need to use a carbon nucleophile and react it at a carbon electrophile. So here are a few carbon nucleophiles, and electrophilic carbons include a carbonyl carbon and an alpha carbon in a substitution reaction. So here are 4 carbon nucleophiles which we can use to make a carbon-carbon bond. So stop the video and draw the structure of each type of carbon nucleophile. So here are the structures. In the aldol condensation reaction, the enolate ion reacts with a aldehyde or ketone to form a carbon-carbon bond. In the Claisen condensation reaction, the ester enolate ion reacts with another ester to form a carbon-carbon bond. So stop the video and identify what else can an enolate or ester enolate ion react with. Enolate and ester enolate ions are nucleophiles, so it can react with any carbon electrophile to form a carbon-carbon bond. This includes an alpha carbon in a substitution reaction and a carbonyl carbon in an addition reaction. So let's practice. Omnia is a household cleaning solvent, so stop the video and propose an efficient synthesis of Omnia from this compound. Note that Omnia has 2 more carbons than our starting material, so that means we have to make a carbon-carbon bond. So the starting material is an ester, so we can make an ester enolate ion, which is a nucleophile, and then react it at a compound with the carbonyl carbon, which is an electrophile, to form our product. In organic synthesis, it's often desirable to alkylate the alpha carbon. So one way to do that is take the enolate ion and react with an alkyl halide in a substitution reaction. But one problem is that elimination competes with substitution, and as a result, the yield is poor. So is there a better way? So there is a better way. The malonic ester synthesis lengthens this carbon chain by 2 carbons. The acetoacetic ester synthesis lengthens this chain by 3 carbons. Each method involves removing a proton from the alpha carbon to form a nucleophile. So the acetoacetic ester synthesis is used to alkylate the alpha carbon in aldehyde or ketone. The malonic ester synthesis is used to alkylate the alpha carbon in an organic acid. In the first step, this compound reacts with a base to form the nucleophilic ester enolate ion, which then reacts with an alkyl halide to form the carbon-carbon bond. In the next step, note that an ester is converted to an acid. And in the last step, a decarboxylation reaction occurs in which a carbon-carbon bond breaks to form a smaller molecule. And again, note that the alpha carbon has been alkylated. Another synthesis strategy is to break a carbon-carbon bond to make a smaller molecule from a bigger molecule. So ozonolysis breaks a carbon-carbon pi bond and makes either an acid or a aldehyde or ketone. A decarboxylation reaction occurs with a dicarboxylic acid with 1 carbon between the acid groups. For example, malonic acid undergoes decarboxylation to produce acetic acid and CO2, so stop the video and identify which bond breaks. So the answer is decarboxylation also occurs with a beta keto acid. So remember that this carbon is the alpha carbon and that carbon is the beta carbon. So stop the video and draw the structure of the product of this decarboxylation reaction. And the answer is, so let's use the acetoacetic ester to make a carbon-carbon bond and a decarboxylation reaction to break a carbon-carbon bond to convert this compound to this product. So stop the video and draw the structure of the intermediates going from the reactant to the products. In the first step, sodium methoxide is a strong base, and that makes the ester enolate ion. The ester enolate ion is a nucleophile, so this can react at the electrophilic alpha carbon in this compound. In the third step, H3O plus is used to convert the ester group to an acid group. Note that this compound is a beta keto acid, so by heating this compound, it'll undergo decarboxylation to form the product. Note that the acetic ester synthesis lengthens the chain in this compound by 3 carbons as mentioned earlier. In biology, there are many reactions in which a big biomolecule breaks down into smaller biomolecules. For example, glucose undergoes glycolysis in a 10-step reaction sequence to form pyruvate. Look for the glycolysis video which shows this 10-step reaction sequence.